the work that we've been doing over the last few years, thinking about a membrane curvature generation. Before I get into the specifics of it, broadly, I want to give you a sense of what my group thinks about. We are theoretical computational folks. We are generally interested in problems that span a whole class of cell shape, cytoskeleton, organelle, signaling, you name it. And we just love all problems that involve geometry changes of various sorts. And today I'll be talking to you specifically about trafficking of particles on the surface of the membrane. And the reason we actually like these problems of cell shape and function is there are classic examples that I could give you. This audience knows quite well of red blood cell shape and function, and sickle cell anemia is an often used and abused example of how change in a certain gene sequence affects the hemoglobin, which becomes sticky, and then you lose the cell shape regulation and oxygen delivery. Other examples, of course, include how cancerous cells seem to lose their shape. We have looked at this over the years in the context of generally cell shape and signaling, and also more specifically in the context of dendritic spines and in neurons, and how these shape affect signaling cytoskeleton features. So these are all sort of broad themes in my group. Today, I'm going to talk to you more specifically about biological membranes. So if you are not familiar with cell membranes, they are self-assembled structures with amphiphilic molecules. So the lipids in here are amphiphilic molecules that self-assemble with the hydrophilic head groups and the hydrophobic tail groups, and they are embedded with a wide variety of channels and proteins and things like that. And as you probably are well aware, washing hands with soap is a good detractor for viruses, simply because viruses have this lipid bilayer that encloses their RNA, and fatty acids break up fatty acids, and that's basically the oversimplified physics of membranes. So one of the things that we got fascinated by for many years now is how these membranes can change their shape. And there are many mechanisms, there are many physical underpinnings, there are many mechanical theories of this, and this has been a very ripe field, very rich, very well developed for the last nearly 50 years now, since the early papers from Canham and Helfrich came out. But over the years, our experimental colleagues across the world have demonstrated how different mechanisms can generate this curvature. So for example, you have lipid asymmetry, you can have protein scaffolding, and how, for example, scaffolding is different from crowding of proteins on the surface. So even though this is a very rich field and a lot of work has been done, we found that there is room for us to make some contributions as well, particularly when we cast some of these problems in the context of experimental observations. And this is basically the series of stories that I'm going to tell you today. So the first sort of vignette I'm going to talk to you a little bit about is thinking about vesiculation in clathrin-mediated endocytosis. So if you're not aware of what is clathrin-mediated endocytosis, this is your Cliff Notes version without all the molecular components. So you have basically the plasma membrane of the cell in response to some cargo event that we don't actually get into the details of here. You have coat proteins that bind the membrane and generate a curvature. Subsequently, actin is known to be involved in this process to generate forces. The final event is that this membrane forms a bud and then undergoes scission, which is outside the realm of continuum, and then this early endosome uncoats, moves down the endosomal pathway and things like that. So what we wanted to think about are some major questions that were open in the field and are still, I think, a matter of some debate. Primarily thinking about what are the interactions between curvature generation and membrane tension. How is force generated around this endocytic pit? And then what are the interactions between membrane tension and actin organization? Membrane tension, as benignly as I have put it here, has also been the topic of great discussion and debate for many years now. And you will see that how we interpret this quantity mathematically and physically can lead to some confusion, and we try to shed some light on it, but these are not trivial problems. All right, so let's get into it. So how do we think about interactions between membrane curvature generation and membrane tension? So the first thing to ask is it even an important question? Should we care? What evidence do we have that this is important? So the evidence that we have kind of goes like this. In cells, it has been historically hard to measure membrane tension. In mathematics, if you think of the membrane as a thin elastic shell, then this so-called tension is simply a Lagrange multiplier for area and compressibility. So how can we get at whether this is important or not? So these experiments that were published a few years ago actually caught our eye because they do something very clever. They take GUVs, vesicles, purified systems, and in vitro reconstituted systems, and they have PIP2 on the surface. It's a lipid required for the binding of endocytic related proteins, in this case AP180 and clathrin. And you can see that these merge, and then you see these curvature bud-like structures. And then they did something which was very clever, which is they changed the tonicity, the pressure difference, by changing the osmotic pressure, by changing the concentration difference of the solute in and out of this membrane. They changed the tonicity, and you can see that if you have a hypertonic condition where you have more pressure on the inside, so high tension on the membrane, you don't see any budding. If it's isotonic, you see some budding. And if it's hypertonic, you see more budding. So it seems that tension is important to form these curvatures. And so we actually set out to think about this conundrum, and then a few other experiments that were in the literature talking about membrane tension. So here is what we did. We assumed that the membrane is a thin shell, incompressible shell. And then we wrote down a bending energy for the membrane. This is simply the Helfrich energy, where this energy density is given by the bending modulus K, the mean curvature squared, except now the mean curvature is offset by the spontaneous curvature, which is the curvature induced by any external moieties, and then the Gaussian curvature. There you will find tons of papers that minimize this energy. You can do this using some sort of you can integrate it and minimize it. You can write your local stress balance. And I personally am a very big fan of partial differential equations. So we tend to write a stress balance and then obtain the partial differential equations that go with this. What you're looking at here is the normal force balance, which gives you the surface Laplacian of this mean curvature. This is now the pressure and then the capillary effects. You're looking at the Young-Laplace equation. And then because you're writing a force balance, you can write any normal forces due to actin. When you do this in this form, and now if your spontaneous curvature is also coordinate dependent, you have to talk about the spatial variation in the tangential plane as well. And here is where things get interesting. Now you have what is known as the gradient of the surface pressure is balanced by any gradients induced by the spontaneous curvature and then the force-induced variation. There is some amount of theory that goes behind all of this, which we have written separately, but here we just sort of used all of that theory and ran simulations in a format where we assume that there is an axis of symmetry, and this is our membrane. We prescribe the boundary condition lambda as the tension at the free end, And then here we describe continuity conditions. And now we figure out if we can actually get at some of these tension effects. So at R is 0, you have the axis of symmetry. The blue region is where we prescribe a protein coat. This is a simulation with high tension, and here's a low tension. Now, what you're going to watch is a movie, but each frame is in mechanical equilibrium. So what is stepping forward in what appears to be time, there is no time here. It's just basically each value of the coat curvature. So if you watch this, as you can imagine from your intuition, you know that when the string is very stiff, it is not going to bend very well and form a bud. Whereas if you have a slightly floppy string, you will see that it forms a nice bud. So far, so good. High tension gives you a sort of shallow invagination, not very good for endocytosis. Low tension gives you a nice bud. And the reason we look at the same values is the same amount of external work you're doing it on the system. So Julian, the student who was working on this at the time, started basically playing with that boundary condition parameter, right? And so we came across something quite interesting at an intermediate value of tension. I'll let you watch this movie, and so you can see what we actually find. Same range of spontaneous curvature. And go. So basically what you see is a snap through instability, which is like if you've played with those frogs or poppers. I actually had one on my desk, which I can't find right now. You played with those poppers. It's literally, there is a small energy difference in this previous state, and then it in the sort of almost bud state, and then it kind of jumps into the lower energy state, if you watch, into that closed neck. So this was actually very interesting, and it actually gave us the chance to think through the space of the spontaneous curvature. The curvature induced by the coat proteins is of a wide range because more than 60 proteins are involved in clathrin-mediated endocytosis. Membrane tension in cells also goes through a wide range, and yet endocytosis is robust. So it appears that if you purely have high tension, you will always get open buds because the system has to do a lot more work to form and close the bud. If you have low tension, you will almost always get closed buds. And then there is this nice intermediary region where the tension and the spontaneous curvature kind of have a competition. You can jump from one to another. And there is this energy barrier. And there is a scaling that you can find that gives you the relevant length scale of the problem. So insofar as what I've shown you here, though, I've assumed something about axisymmetry of this geometry. And then I'm telling you there is this energy barrier to go from open to close. So what happens if you let go of the assumption of axisymmetry? From a practical standpoint, what happens if you let go of the assumption of axisymmetry is now you're no longer solving your system of PDEs as a boundary value problem along an arc length, which you would do here. You're literally solving a system of ODEs with boundary values. You now have to solve the entire surface parameterization. You're getting into issues of meshing, you're getting into issues of PDE stability, finite element methods, and like I tell most of my group, the line between brave and foolhardy is very, very thin. And sometimes such projects cross this line very often. So this was actually a very brave undertaking because it worked. By Ritvik, my former student who's now at the Allen Institute of Cell Science, and our collaborators, Shiva Rudraraju and Krishna Garikipati, who actually developed this code using IGA, isogeometric analysis, excuse me. And what we found was actually fascinating. So as before, if you take your, this time we're actually looking at how to pinch the neck. So if you start with an axisymmetric model, and you try to pinch this, so you're applying a force and closing it down to form the neck, you get a snap-through instability at these regions that have this negative Gaussian curvature. However, if you take the same geometry but do not impose an axis of symmetry, in fact, you solve it on this entire geometry, what you end up getting is not a snap through instability, but almost the ease of constriction, the ease with which you can form this neck and close it is much more than what you would if you had imposed axisymmetry. The analogy for a lot of these geometries that you're seeing is if you crush a soda can, if you just crush it, that's essentially the geometry that you're seeing. So even though we noted that there is a snap through instability between tension and spontaneous curvature, we also note that if you let go of certain restrictions of symmetry, you can actually get what is known as a near spontaneous curvature, a near spontaneous constriction. So for endocytosis, you actually don't need to do very much more work to close the neck once you are able to reduce the degrees of symmetry. And you can actually extend these ideas to thinking about helical structures of something like a dynamin, and how these dynamin-like structures can sort of form, and how the pitch of the dynamin structure can lead to these different constrictions. And what I can tell you here is that as you go from a zero pitch to an increasing pitch, it is easier to constrict the membrane. In all cases, because we have no imposition of symmetry, you immediately get the reduced symmetry mode. And so if you go back and look at the proteins involved in endocytosis in cells, letting go of symmetry, allowing for helical forces, they all actually make constriction easy once you form the bud. So this was essentially our sort of first foray into thinking about how we can interface tension, forming a neck, pinching the neck, and things like that. But then there are some cases in which you can actually look at the neck itself as an object that seems to be quite stable. So for example, if you look at clathrin-mediated endocytosis, we've talked a little bit about these helical structures like dynamin. But if you look at these caveolar structures, caveole in complexes form, but it's a pretty darn stable neck for prolonged periods of time. And so we got interested in thinking about this, and this was work done by my former postdoc, Morgan Chabanon, who is starting his own group now this month in Paris. And we were wondering if there are simple relationships we could extract between these neck structures and the protein distribution in there. So as always, we kind of go back to the experiments to see what we can make sense of and how long these stages of these necks form. So what you're looking at here is EM work from Ori Avinoam, who has very gracefully shared some of this unpublished work with us. So you can see the plasma membrane, early bud, formation of a U-shaped bud, and then formation of this nice neck-shaped structure. It's not a very narrow neck. It's a pretty nice wide neck. But you can immediately see the shape of a catenoid here. And then subsequently you have scission. So Morgan essentially mapped out from these structures the mean and the Gaussian curvatures, and identified that there is a fairly nice regime in which you can approximate the neck to be a catenoid. A catenoid is a beautiful, beautiful structure geometrically, because a catenoid has zero mean curvature everywhere. The two principal curvatures are equal and opposite, equal in magnitude, opposite in sign, and the Gaussian curvature is negative everywhere. So now you can think of this mathematically as the inverse problem. Previously, I said we have the PDEs, we're going to apply an input and solve for the shape. This time, I'm assuming the shape and asking what solutions do the PDEs allow. And what I'm looking for is what are these spontaneous curvatures, which I can interpret as the protein distribution that are allowed along these catenoid necks. So you can think of this as there is a bigger bud somewhere, there's the rest of the cell membrane elsewhere, and what are allowed. So if you start with the same spontaneous curvature as if there were a reservoir of proteins. So here's a good example. If you had a reservoir of proteins on top and a reservoir of proteins on the bottom, and you have the same boundary conditions, what we find is as you go along the Gaussian curvature, this is the map of the Gaussian curvature, the spontaneous curvature follows the Gaussian curvature. This is negative and increasing in magnitude. This is positive and increasing in magnitude, such that to maintain the catenoid shaped neck as an energy minimizer, you would need to have a non-uniform distribution of proteins in the space. And now you can extend this argument to looking at what happens when you reduce the radius of the neck, because remember, in constriction, we're really trying to get this neck as small as possible. So there are certain radii above which you get spontaneous curvatures that continue to follow the Gaussian curvature, but are of a certain sign. And when you reduce the neck size below a certain radius, what you see is that you have to, the only way to accommodate this neck, this small neck, as a stable membrane structure is to flip the sign of the spontaneous curvature. In other words, a simplified way of thinking about it is if you had cone-shaped proteins that were going along here, and as you could maintain the same cone-shaped proteins, different distributions here. But as you decrease the neck, you would need a second class of proteins that were with this cone reference point wedge-shaped on the side that would help maintain the smaller neck. And that can be energetically expensive. If you don't have access to that, then it is cheaper to just fission off. So you can map the space of the spontaneous curvature and the neck radius, non-dimensionalized obviously, and find that there is a critical, so this is basically just a parameter space of the same map I showed you earlier, but here you can see that there's a critical energy barrier for a critical neck length. And to go lower than that, you would need a second class of proteins to maintain the spontaneous curvature. Are there ways to tune this energy barrier? Obviously, we use the fact that there are so many different classes of proteins and cells on the membranes that we can sort of play with this a little bit. So now I have two different boundary conditions on top and at the bottom, and I play with these values of C1 and C0. And if C1, and for a majority of conditions, you get this energy barrier. But then there are some nice sweet spots, like when C1 and C0 are equal and opposite, where actually you can keep maintaining a smaller neck with no divergence in the energy barrier. It's like there is a nice smooth transition as the neck gets smaller and smaller, and you can look at this energy diagram and see that it doesn't actually blow up. It's 1 point on this space. So essentially, the boundary conditions determine the location of this energy barrier, but so does the height of the catenoid. We simplified this catenoid to be a nice equal catenoid on both sides, but you can imagine that there are, half catenoids or various degrees of asymmetry. So a good example is what happens on a tube is basically you have a half catenoid and the rest of it is a cylinder going up from top. So again, if you sort of lop off different extents of this catenoid, you will find that if you have basically a half catenoid, there is no energy barrier. This is actually what this half catenoid is where you would stop at this dashed line. And this is actually what you would see in tubes, membrane tubes as they form. Okay. So basically thinking about spontaneous curvature as a way of membrane protein interactions, you can basically simplify the problem, look at different necks, and see that a whole variety of shapes, you can obtain them that sort of span the expanse of these various necks that you see in endocytosis. Okay, so that is as far as curvature generation, tension, and protein interaction goes. I'm just going to grab a drink of water. If there are any questions, I'm happy to take them.
So I was going to say, if anyone wants to ask any questions, then you can just type them into the Q&A and you can ask them.
Okay, all right. So I said all this about proteins. There's so much you can do with Helfrich. So what do we do about forces around the endocytic pit? So one thing that we noted, again from experiments, was the following. We know actin is important for force generation in many membrane processes, including endocytosis. But actin is not always required. It is required when there is a high membrane tension to continue endocytosis robustly. So if you have high membrane tension and you somehow do not allow for actin polymerization, so latrunculin to inhibit the actin polymerization, just plecanolide to stabilize the actin filaments. In both these cases, you can see this big wide neck and it's a stalled endocytosis. So if you leave things well enough alone in high tension, endocytosis is still robust. So how do we think about this role of actin? So we can do the simple thing. We can go back to our continuum model. You will recall that I said because we write normal and tangential balances, you can just add a force, treat it as a free parameter, and then run your calculations for shape. And we did that originally. And one of these sort of things we were trying to get at is the actin is exerting a force up and down, or if it's sort of exerting this collar-like constriction force that we were looking at a little bit earlier. And what we found was that in all the cases of those high tension simulations that I showed you, if you apply forces which are not, they're just applied in here as a parameter, but they are ascribed to actin, you can of course have a better progression of endocytosis. The physicist in you will argue, well, that's not a surprise. If you put more and more work into the system, you can make more progress. And I completely agree with you. The question is how to think about it in a manner that is more considerate of what we know of actin dynamics. So this, again, a very brave project by Matt Akamatsu, who is a postdoc that I co-advise. He's at UC Berkeley, and I've been working with him very closely from the beginning of his postdoctoral days. He's with David Rubin and works with me and him. And he basically said, we're going to build an agent-based model of actin interactions with the membrane. So essentially, he went through, found all the known rates, to the extent that locations of various molecules involved in actin are known, he found them and found the key pieces of the puzzle. We needed to know the ARP2-3 location. They're the base of the endocytic pit. The attachment of the actin to the tip of the pit is through a linker protein known as HIP1R. And he set out to develop this multi-scale model. The first thing we did was we made a simplification of our entire membrane mechanics, where he essentially took the force versus the internalization curves from the previous models I showed you and found the linear regime and used this linear regime to calibrate a spring for the membrane. So essentially he took this force tension curve and used that to build a spring constant where the endocytic internalization is essentially proportional to this membrane tension. So he took that and then he did something else that was phenomenal. He went and counted the number of ARP2-3 molecules in experiments. These are methods beyond my ability to explain, so I will not even try. But essentially, he found that there were roughly 200 molecules of ARP2-3 at the base of an endocytic pit. And now in our model, we know all these parameters. We have the number of ARP2-3. We modeled actin using cytosine. And here we simplified the membrane to simply be an elastic spring. So we are good to go. And now I have to tell you this though. So we have a sphere with a spring. So what you will not see here is the deformation of the membrane. What we are looking for is the internalization of this endocytic pit as a function of actin polymerization. So everything here is parametrized. Now what you're watching is a preformed pit. These are the HIP-1R linkers in purple, violet, purple. This R23 in blue. The actin nucleators, you can see the growing ends, you can see the capped ends, and the white are the actin filaments. So this is an example of a simulation where the actin is now undergoing growing, branching, severing, and all the reactions that one expects actin to undergo. And you have watched the pit, the tip of the pit, move inwards, internalize quite nicely in response to this actin remodeling events. And you can track the internalization as a function of time. You can get an internalization velocity. And we set this to a high tension regime. So you can essentially see that, yes, actin polymerization can counteract these membrane tension effects and internalize the pit. Now, from simulations like these, you can also extract some nice points to kind of interact better, I think, with your experimental collaborators. So this is actually the color. What you're looking at here are the number of actin monomers around the endocytic pit. The color scheme is deliberately chosen, very deliberately chosen to look like a super resolution imaging of actin, because we had a colleague who was actually looking at these and, you know, they look similar, sort of thing. But now you can actually start to ask questions about, are there any higher order principles of actin organization around this endocytic geometry? So you can look at the orientation of the actin filaments, and you will find that around the pit, majority of the filaments are oriented up and down, and you can see that here. But as you go away from the pit, a majority of the filaments seem to start to bend towards the base of the membrane, and you can see some of these quite dramatically here. The other thing you can look at is the radial orientation, and there is really no preferred radial orientation. So even though we have a stochastic simulation, after a long enough time, you see a nice radial distribution of actin, and then there are these bent actin filaments that, if you look in the ZR. Okay, so what are some of the things that we can think about and take away from here? One thing we noticed was these really nice bent filaments, and we saw this again and again. In fact, if you're able to see my mouse and you can follow this, you can see this little guy stapled here with 1 linker, 2, 3, 4, 5, and then it kind of bends outward and goes this way. So we're pretty sure this is too good to be true, right? It must be an artifact of how we set up the simulation. And then I have to tell you that Daniel Sarwas, who is stop doing that. Sorry. Let me pause this. Okay, so Daniel is a cryo-EM postdoc with Drubin, and we were discussing this result, and he actually offered to share with us his cryo-EM work around an actin pit, where the membrane here is the this is the plasma membrane, excuse me. I'm marking the clathrin-coated pit for you. These lines that are appearing are actin filaments. And in fact, if you watch as the membrane deforms, you can see these sort of long bent actin filaments around here. So this was actually a nice sort of serendipitous event where you are watching these simulations predict bent actin filaments, and we have this little piece of evidence that such bent actin filaments may also be present in cells. And ongoing work right now is where we are analyzing with Daniel a lot of these tomography images to make sense of the actin organization. There's a lot of, as you can imagine, image analysis type work involved in there. But seems that endocytic actin bends around the clathrin-coated pit to promote this. And one of the things we predicted in this work is what is the sort of point of bending? You know, how does it help? So if you think about the energy that is stored in the actin polymerization, the system that I showed you, versus the energy required for the PIT internalization, you only need like 2% of this, all this actin work to internalize the PIT as opposed to what is stored in the polymerization. Filament bending stores about an equal amount of energy as you need for internalization. So we actually showed this in the paper where we said, okay, what if you suddenly dramatically increase tension? Or you cause a dramatic change in tension. And in that case, these bent filaments almost like let go and provide a force to internalize the PIT even stronger. So they seem to have some sort of a buffering capacity to internalize these endocytic pits. Okay, so that's two segments. We talked about curvature, we talked about actin. Then can we start to put these pieces together a little bit more cohesively rather than doing them on a piece-by-piece basis? So let's see if we can do that. What can we think about the interaction between tension and actin organization? First of all, using this agent-based model, we can reconclude that if you increase the spring stiffness, which is now this parameter in the model, you will decrease the extent of internalization. So high tension, even when you have actin filaments, really high tension is not conducive for internalization. If you just look at varying the ARP23, which is the branching protein, and its nucleation rate, you get essentially some benefit of increasing ARP23 nucleation rate, but not a very large benefit after some time. You get some sort of a saturation. Similarly, if you change the surface density of ARP23, you don't get a very dramatic effect. However, if you combine the two, you now take the spring stiffness and the ARP23 nucleation rate, you can see that if you have very little nucleation, you're going to have very little internalization at various tensions. But even at high tension, if you now are able to provide a larger actin nucleation rate, you can get better internalization by this coordination of ARP23 and tension. And so Matt actually did a very clever experiment to show that this was indeed possible by looking at the lifetimes of clathrin and dynamin, the two key proteins involved in endocytosis, in in SKMEL2 cells, which are known to have high tension. And what he did was he used a compound named CK666. This is a pharmacological inhibitor of ARP23 activity. So if you're increasing this concentration, you're decreasing the ARP23 activity. And increasing lifetime of these components means reduced endocytosis. So in these cells that already have high tension, you can see that if you decrease the ARP23 activity, you are inhibiting endocytosis. And you can do this also in a time-dependent manner. So to the extent that we can look at some of these interactions between the actin-dependent proteins and the membrane tension, there is a certain coordination between the filament nucleation branching activity and membrane tension to promote internalization. Then the last piece of this puzzle that we want to tease out, so we talk a lot about what happens at the plasma membrane, ARP23, counter numbers of molecules, and things like that. Unlike cell motility and problems at a leading edge or lamellipodia, endocytosis has a further complication. You see, it's not enough to polymerize actin here. You also have to catch and link it here. So you can think of this as a problem of two boundary conditions. So what happens if you change the spatial organization of these linkers of actin in here? So this is actually very hard to do experimentally, to change spatial localizations of molecules, but we can do this in a simulation. So we did the following. We took the same numbers of HIP1R, and put them all at the tip here of the pit, and distributed them increasingly across the surface. And this is the condition that you have seen before. Oh, they all run. Let's pause them so we don't get confused. Oh, I don't know how to do that. Never mind. So you can see that in this case, it's not productive. You have actin polymerization, and it's not productive. In this case, where you have some distribution, you can see immediately the filaments bind and bend, but the internalization is nowhere nearly as dramatic as it is in this sort of control case that I showed you, and this is somewhere in between all these cases. So you can actually go in and determine the sort of internalization velocity of these different conditions, right? So if you have just a 1% coverage to an 80% coverage on the hemispherical tip, and what you see is that you need at least a sort of 50% coverage based on our parameters to have a robust internalization. So you see this regime here, they're all starting to overlap quite a bit. And when you have this sort of coverage, what you end up getting are sufficient number of barbed ends at the base, that's the R23 port, and then sufficient number of R23 that are actin that are not R23, actin that are caught on the tip that can hold on, they're linked, and then you also start to see this increasing bending. So basically, it's not sufficient to just think about R23 at the base of the membrane. You also have to think about the HIP-1R coverage and how this internalization is affected. It is proportional to the coverage, surface coverage, and the number density of HIP-1R as far as actin internalization goes. And so to summarize a lot of these ideas of thinking about actin membrane interactions and tensions, you see these nice bent filaments. Matt can count molecule numbers. We are very fortunate to have experimental collaborators who share these kind of data with us and go off on these experimental method developments to do this. You have actin that self-organizes into this nice array. You can think of majority of these forces are orthogonal, and then you have, if you want delayed force production, you have this sort of bent filaments that are very useful. So in all of this, you can kind of come to some conclusion about how self-organization at two locations, using two boundary conditions is useful. Now, I just want to, this is pretty much the bulk of what I want to talk to you guys about, but I want to throw out some sort of newer ideas there for us to ponder and maybe discuss. Even though I move around the HIP-1R, I prescribe these distributions, I actually never told you how proteins may diffuse on the surface of the membrane. We've thought about it. It's, as you can imagine, mathematically not trivial to couple the curvature generation of the membrane with proteins. And this work actually just came out last week, where we developed the entire theory of coupling diffusion of proteins on the surface with curvature, with your Helfrich elasticity, and lipid flow, which controls the what we're calling membrane tension, but what is really the surface pressure. So you can watch, for example, if we initialize the system with two patches of proteins in here, over time they diffuse, the purple tends towards the green, and they nicely diffuse away. We can simulate this sort of coupled deformation. This movie here is actually even more interesting, because we start with the formation of a bud. You can almost think of it as a fusion of a bud onto the membrane. And we have high protein density here. If you watch this when I run the movie, you will see that for the neck, which I've gone on and on about for a good portion of the past hour, for the neck to sort of release and open, this tip will actually come down. So you see it elongates and then it diffuses, which is actually very cool. We'd never seen something like that before. So now we have some of the technical parts worked out. Then the other part is, of course, all of this is continuum. How do you deal with the sort of discrete nature of these proteins? Just we're by training, I am a partial differential equations, finite elements kind of person. I'm only just getting into these sort of coarse-grained simulations. What you're looking at here is a lipid bilayer with nth domain proteins containing epsilon nth domain, trying to move around and diffuse. I am trying to wrap my head around these primarily with the goal of how can we better parameterize our continuum models and bring them closer to experiments. This is just a sort of pitch for some of the other things we've thought about in this membrane protein world. If you are very inclined in sort of the mathematical complexities of how to actually solve these equations, we've been doing some work on sort of sensitivity analysis, which parameter to choose, why does my simulation break? We've been thinking about that quite a bit. And of course, our other kind of fun thing we do is we take 3D reconstructions from electron tomography, and we are actually trying to quantify these curvatures in realistic geometries. How important is curvature? This is the curvature, the two principal curvatures on a dendritic spine and a dendritic spine head. So this gives us a sense of how well can we approximate how close to a cylinder is this and things like that. And with that, I will thank all the people who do the work. So these are my Berkeley colleagues, Julian, who is now graduated, Matt, who is an amazing postdoc who's on the job market, David Rubin, my experimental collaborator for all things endocytosis, Shiva and Krishna for working with us on these sort of finite element methods. And this is my group back when we could hang out in person many moons ago at UC San Diego. And I will put in a shameless pitch for if you kind of want to join us. We do have 2 positions open, so please email me. And I think that's it. That's all I have. Thank you.